Well, thank you very much, Michael, and it's really a pleasure to be here, even if you don't like my title, you know. We have so much fun in debating, and I think that research is all about that, having different controversies, but staying friends. So that's a good thing about research. So within the next 25 to 30 minutes, I would like to give you an overview, an introduction. I think that Frank Diehl has already introduced this term, but I think I can probably give you a little bit more of, insights into also clinical validation of these new analytes. Well, we introduced actually the term liquid biopsy, and that's why we like it actually, about 8 years ago, and in a journal that was called Transmolecular Medicine, and we had at that time no idea how popular it would become. It's almost a buzzword. And at the last AACR meeting, there were two main topics, which was immunotherapy, of course, and liquid biopsy, and I had the pleasure to give this open plenary talk in front of a few people, and this is really my picture together with the head of the AACR, which is March Foti. So what do we mean if we say liquid biopsy or liquid profiling? What we actually mean is that we analyze either cells or cell products, and among the cell products released from the tumor cells, the DNA, of course, is the most prominent target. What is the rationale? Well, pathologists are not out of a job soon. I have no doubts that that is true. I think that tissue biopsies are still the gold standard in diagnostic, but they have some limitations. They are invasive, and some locations are difficult to access. We also know if you just get the biopsy of one metastatic site, you may miss other genomic alterations in other metastases of the same patient, so there's heterogeneity. And also you cannot sequentially take biopsy every 1 or 2 weeks during therapy, so taking blood samples also has the potential to do a real-time monitoring what's going on during therapy response. So the vision is to get more comprehensive and more real-time information by the analysis of blood or other body fluids. Well, as you can see here, it's really an increasing interest in these fields. These are the two main analytes of liquid biopsy, CTCs and ctDNA. And you can see here there's an exponential increase in the number of publications for the circulating tumor cells here in blue and for the circulating DNA here in orange. But I think it's also very important to notice that it's not only publications that are going on, but also clinical trials to prove clinical validity or utility of these analytes. And as you can see here, there are more than 350 active trials using CTCs as biomarker and more than 200 trials using ctDNA as biomarker. So what are the clinical applications that are actually analyzed in these clinical trials? Well, what can we use it for, with other words? Well, there's of course the dream to detect cancer earlier, so to use it for diagnosis, and I will make some remarks on that. There's also the wish to actually do a follow-up and see how actually the therapy is working in a patient, so to predict actually the response of therapy. There's also the wish to get more information on tumor evolution, how does therapy actually affect the tumor cells, and what kind of mechanisms of resistance occur during therapy, and how can we overcome those. And last not least, there's more and more interested in this stage of minimal residual disease, which is well characterized in leukemias, lymphomas, but poorly characterized in solid tumors. And that means actually the stage when surgery has taken out the primary tumor, but there are still some occult micrometastases that can now be detected with these sensitive methods and may predict which patients will relapse. Well, for the circulating tumor cells, we know already that they really can undergo some significant phenotypic changes from epithelial to mesenchymal transition. We know that they occur as single cells or cell cluster, and most importantly, we also know that they are different in terms of the phenotype and genotype. Not all of the cells in the circulation that are derived from the tumor have the same potential. And of course, one of the key questions is to identify the cells that can home and initiate metastasis in a secondary organ. The key challenge for the last 10 or more years was actually to develop assay that can detect tumor cells in the background of millions of normal blood cells, and that's now doable. And the principle of these assays are that we first always do an enrichment and then a single cell identification. I don't want to go into any details, just to show you one example of a chip that we are developing in Hamburg in the context of an ERC grant. Here we have actually a chip with strapped avidine dots. So we incubate our blood preparation with biotin-related antibodies to cell surface antigens, for example, EpCAM, HER2, or EGFR in the case of carcinomas. And then actually the cells can actually be captured on the strapped avidine dot through the binding to the biotin ligand. We can take off the lid of the chip like a cabrio and then pick actually single cells for further molecular analysis, or just throw the whole chip into a petri dish and then culture the tumor cells. Well, for the molecular analysis, of course, we invested quite some time to figure out how can we do a whole genome amplification, because we need to amplify the DNA to have enough for NGS and other methodologies. And there are also some cool, I think, instruments around where you can do the single cell sorting automatically and get cells with a defined phenotype. In addition to this descriptive analysis, it's now also possible to do functional analysis on circulating tumor cells, either using xenografts, and we published together with Andreas Trumm from Heidelberg the first xenograft assays from CTCs in patients with breast cancer, or we can also establish cell lines. Besides circulating tumor cells, circulating cell-free DNA has become a popular analyte of liquid biopsy assays. And as most of you know, and it was already introduced, the DNA is majorly released from necrotic or apoptotic cells, but there might be also a mechanism of active secretion, and there are all kinds of features of the DNA that can prove that that's derived from the tumor cells. And as Frank Diel has already pointed out, again, we have here the situation that we have a low concentration of tumor DNA in a high background of non-tumor DNA. So we need very sensitive methods going down below 1% of tumor DNA that can be detected in the blood. So we need highly sensitive technologies. And we have to be careful because we are all getting older, unfortunately. And if you get older, it happens what you can already read in your textbook when you went to high school, which means if you get older, you acquaint more and more tumor-associated mutations. Most of those cells just die. They never form a tumor. But with the sensitive technology, you can now actually detect these tumor-associated mutations, such as hematopoietic leukemia driver mutations, P53, or KRAS mutations at low level in individuals that have no sign of cancer. So we have to be careful if the sensitivity becomes too sensitive. Like with every technology, it's not really surprising. So what are the clinical applications? Well, the first thing is we can see, is there relevance in early stage cancer patients? Here these patients have been operated by surgery, and the key question is, do the patients still have a risk to develop metastasis? And we know now from many, many studies in different tumor types that the number of CTCs in a blood sample at the beginning of or after surgery is a very good indicator of an unfavorable prognosis. So it seems to be, if you find intact tumor cells in the blood, it seems to have an important impact on the outcome of the patient. So that could be used for improved tumor staging besides the lymph node analysis. We also have done that in patients with breast cancer that receive neoadjuvant therapy. So they get their chemotherapy before surgery. And we could actually show that if we do that, the CTC count provided information which was independent from the response of the primary tumor. This response of the primary tumor is a gold standard in neoadjuvant trials, so it was important to show that if you look at the tumor cells in blood, you get additional information, and that has been also now confirmed on more than 2,000 patients with breast cancer. So this is from the confirmatory trial. You can see here that you have an increased risk to develop relapse if you have a higher count of your circulating tumor cells, but also look at this ratio. the risk to develop a relapse was more than 6 times increase if you had a higher CTC count. So quite a strong predictor of relapse. The other question is, if we do that at surgery, that's okay, but then can we also do that in the follow-up? Can we get later on also blood samples to see which patient has a higher risk to develop relapse? And that can be done too. That is also the publication that we did together with the German breast cancer study group. And here we actually took the blood samples 2 years after the initial chemotherapy and surgery, and we could show that the CTC count was also a predictor for a worse outcome. So I think probably there is a need in the future to do trials not only at the time of surgery or at the time when the patient has already a metastasis, but also do clinical trials in between this period and use these kind of analytes to predict which patients have the highest risk. This can be also done by circulating DNA. This is a very nice publication here, published a couple of years ago, and they measured circulating DNA in patients with breast cancer. At surgery, there was no difference in the outcome, but if they did it after surgery or during the follow-up period, there was a clear separation between the patients that had ctDNA and had not ctDNA, but you also have to make sure that here the number of patients investigated were rather low. Well, how can we go on? I mean, we also want to get more information besides enumeration. Can we get information on the characteristics of the DNA or CTCs that are in the blood? And here, the key focus from the clinical perspectives are therapeutic targets, such as HER2, estrogen receptor, or PD-L1, or mechanisms of resistance. And I just want to show you one examples for the circulating tumor cells. Here, actually, the group of Emanuel Antonarakis published a few years ago already that if the prostate cancer cells express this antigen variant 7, then they can become resistant to anti-androgen therapy with these agents, abiraterone and enzalutamide. And the question was, can we also measure that on the CTC level? And in this publication from last year, he could show that patients who had no CTCs anymore after the therapy, the response was very good. The PSA levels all go down. If the CTCs were still present but did not express the resistant variant, the response was still not too bad. But if the CTCs were present and expressed the resistant variant, then most patients had an increase in PSA, so no good response to that therapy. Of course, it's not only black and white, I mean, expressed or not expressed. You know that tumor heterogeneity is a hallmark of cancer. So what we actually did a couple of years ago, we did actually a multiplex mRNA profiling. We picked single cells and looked at the mRNA profile using RT-PCR, and Michael loves to hear that. We are using RT-PCR, of course, for more than 10 years now, so we are convinced. So here you see the single circulating tumor cells. Here you see the profile of all the transcripts. It's just, I think, some of them, not all of them. But for the ARV7, you can see there is some green and there's some red and there's some green, indicating there is some expression and some non-expression. So this heterogeneity might be important in the context of therapy. Well, of course, what is also very important as application is, can we actually get the molecular evolution during therapy? Can we actually see which kind of mutations are there, and does this composition of mutations change during therapy? And I think here the ctDNA is certainly in the front run. There has been FDA approval on the detection of EGFR mutations in the context of EGFR-targeted therapies in patients with non-small cell lung cancer. And the good news here is the specificity, if you compare the blood to the primary tumor analysis, is very high, almost 100%. So if you find a mutation in the blood, it's also present on the tumor. But the sensitivity in this original publication was still moderate, with only less than 66%. But then the FDA made a very pragmatic conclusion. It says, well, why don't you look first in the blood? And if you find something, then you don't have to do a biopsy. And if you don't find anything, then you may have to go through the biopsy. A very pragmatic, I think, kind of conclusion. And you still see that the sensitivity of the blood analysis is still a bit lower than the analysis of the tumor tissue. But of course, it's a non-invasive procedure. We are also in the middle of analyzing a methodology which is not based on NGS, because NGS always requires bioinformatics, and bioinformatics is not very well standardized in many of these assays. So this is an assay based on mass spectrometry to detect mutations, and that was a publication, which is not really published yet, a presentation where we looked at patients with lung cancer which have brain metastases. These are difficult patients because the brain metastases do not release as many cells and DNA in the circulation, probably due to some problems with the blood-brain barrier. So we took the toughest, I would say, application, but I think we got informative information on the ctDNA in about almost 50%, and the concordance to the primary tumor was again very good. So we could here see some initial mutations, such as this mutation at exon 16 deletion mutation, but we could also find some mutations only in the blood that occurred after the initial diagnosis, for example, this resistant mutation T7090M. So you can also get some real-time information by a blood analysis and may not need next-gen sequencing technologies. Well, I think in colon cancer, Alberto Bardelli has really pioneered the field looking at the development of new mutations during therapy. He actually looked at the mutations before therapy and after therapy, and he could see that a number of mutations come up during the therapy, are probably induced by the therapy. And these mutations can confer resistance. And I think that is really the beauty of blood analysis. You can actually take blood samples here every 2 weeks, and then you can measure the mutations that come up and maybe in the future adapt the therapy to these mutations. The similar thing was done in breast cancer by the group of Jackie Shaw, published here, and she did actually analyze mutations relevant to the resistance to estrogen targeting therapies, such as mutations in the estrogen receptor. And she could really show that before therapy with anti-estrogen targeted therapy, there were no mutations, everything is white here. And then in the 4, 7, and 12 months after initiation of therapy, these mutations arose in these patients. So very nice to see that you can get some acquired resistance, and you can measure this resistance in individual cancer patients. So I think that I've also adapted that to circulating tumor cells, and I think I've shown that before. So one of the most, I would say, popular themes which you find in the newspaper every month now is, can we use this technology for early detection of cancer or screening of large populations? And can we detect cancer early with the premise that early detection might, and I say really might, lead to more cures. This is still an hypothesis. It's not so easy. So one of the key publications was in patients with nasopharyngeal cancer, which is a very frequent cancer type in Asia. And Dennis Lowe has published this last year in the New England Journal of Medicine with a remarkable sensitivity of 97%, specificity of 98%. And if you have a postdoc or doctoral students, please tell him the number of subjects that were screened, right, before he starts his doctoral thesis. So you see an enormous effort. And interestingly, he looked at an Epstein-Barr virus DNA quantitatively, which is not really a tumor-specific target, because we all have EBV DNA, or most of us, in our blood. But he could work that actually out. I think that is really very encouraging. However, nasopharyngeal carcinoma doesn't play a role in Europe or in the USA. So we are, of course, majorly interested in colon, breast, lung, and prostate cancer. And here, this publication from the Johns Hopkins group came out this year, in the beginning of the year. and they claimed that they could really detect now the tumor cells from 8 different tumor types. What they have actually used is a broad panel of mutations on 16 genes. I think more than 1,000 mutations were detected, and that was not enough. They combined that with the detection of 8 protein biomarkers. Most of these protein biomarkers were quite well known, such as CEA or CA99, so there was nothing really surprising in the protein marker. But then they used this machine learning thing, which I never understand, but I think it's probably very important. And I think they really came up with a very high specificity. They had 100% specificity and a very good sensitivity for some tumor types, such as ovarian cancer. So that could be really something interesting for that. For other tumor types, such as breast cancer, the sensitivity was around 30%. And if you look for the sensitivity for early cancer stages, and that's very important because we don't want to detect a large T4 tumor, we want to detect small tumors, then the sensitivity was more in the range of 50% or less. So I think there's still some work to do, I think, in terms of early detection. And the costs for that was estimated $5,000 per analysis. I don't know whether any health system will be able to pay that for mass screening. I'm a bit pessimistic about that. Anyway, we have in Hamburg started actually to create a big biodepository. We have started the Hamburg City Health Study a couple of years ago. We will screen 45,000 Hamburgers between 45 and 74. This is not done in conjunction with McDonald's. These are, of course, individuals living in Hamburg. And as you can see, we get many data, and we will get a few hundred cancer cases, and probably this will be a wonderful biorepository for future studies on cancer screening. So, is there anything beyond CTCs and ctDNA? Of course, there are a lot of activities going on. I don't have time to go into details. Just to mention, there's an enormous effort in the analysis of exosomes. These are small vesicles, extracellular vesicles, that are released by cells and can be taken up by cells. And these exosomes contain everything that the cell contains, including proteins, RNA, and DNA. Interestingly, the exosomes that are taken up can also change the biology of a recipient cell. For example, we have shown with David Leiden that they can actually guide organ-specific metastasis, so it might be an interesting biomarker with a functional activity. The other liquid biopsy biomarker that is also analyzed very frequently are circulating cell-free microRNAs. And as all of you know, microRNAs have various biological functions, and they are released either within exosomes or bound to some proteins and can be detected by PCR technologies in a very sensitive measure in the plasma of blood. And I think if you're more interested in it, we had published a review 4 years ago, and now we have actually published an update on that field together with George Carlin, which is probably one of the pioneers in the microRNA field. Besides that, I think there are also new kids on the block. Here, the work from Tom Würdinger from Amsterdam has shown that tumor-educated platelets can have certain RNA species, and the tumor environment change the RNA composition of these platelets, and that can give also information on the tumor, a very exciting new area. We have started actually in Europe to establish working groups. We have the so-called European Liquid Biopsy Academy, or ELBA, which is headed by Tom Würdinger. Here's a focus on the detection of lung cancer. And we have started a prostate cancer group actually focusing on the detection of high-risk prostate cancer, with the idea to actually not just detect prostate cancer, because every man above a certain age probably will have prostate cancer, but find out which prostate cancer are clinically relevant and deserve therapy. So in order, I think, to really bring everything from a beautiful publication in a wonderful journal into the clinic, we all know that that is the biggest bottleneck. And the EU has granted us a few years ago a new consortium in the IMI network, which was focused actually on looking at the robustness of methods, nothing else, just a technical and a little bit of a clinical validation of methods. And we were very happy that we could actually get this grant started. We are now 38 different partners from all over the EU. We have on board academic institutions, non-profit institutions and companies, and our focus is on lung cancer and breast cancer. So in order to go on with it beyond our funding period, we have also established the European Liquid Biopsy Society as a continuous network of these activities. So what is the philosophy of Cancer ID? I think we have dealt with the technical evaluation of methods. We go in the direction of establishing working protocols, and at the end we'll do a clinical validation to get really robust methods that could be used in future clinical use. For example, one of the activities that we are very much interested in is, can we do a combined analysis of cells and cDNA from one single tube? So I think what we have done is we take the plasma fraction, we take the cellular fraction, and we see how well we can do the genomic analysis from both sources out of one tube. For the clinical validation, we have focused on immunotherapy because it's now the hottest area in clinical oncology, the use of inhibitors of immune checkpoints. And here we will look at the PD-L1 expression, which is one of the key targets on the circulating tumor cells, but we combine that with the analysis of the mutational load, and we can do that at the ctDNA level. Just to give you a flavor, what are we doing in terms of methods in this group? Here, for example, this is our ring trial on ctDNA. We have spiked blood samples with clearly defined DNAs that contain KRAS mutations and EGFR mutations. Then we send these preparations to different partners which actually use their specific methods to extract a DNA, and then the results of the analysis, then all of this is shipped to our partner that performs here a ddPCR to see what are the difference of these analyses used by the different partners. With the Liquid Biopsy Society, we would go on with this activity and create a network not of individual members, but again of institutions that can actually help us to bring these technologies into the clinic, because I think this is really the clear bottleneck. We publish a lot, we have wonderful meetings, I give so many talks, but so little of any biomarkers arrive in the clinic, and we need to change that in the future. Otherwise, I feel that our work is a bit just for ourselves, but not for the patients. So, at the end, what is my conclusion? I think that we don't have to fight which of these analyses is the best. I think they are complementary depending on the application. I think it's very important to validate the assays by independent expert group. There was a very good editorial in Nature Biotech this year showing that if we don't do that, then the public will not believe that these assays are good. And I think it's also important to get clinical utility. So you have to show that your liquid biopsy or any biomarker assay is changing medical practice. What are the clinical applications? That is the key. But if you do that, be careful that your therapy must work. We had a bad experience with the circulating tumor cells. There was a stratification and a change of therapy from one therapy that didn't work to another therapy that didn't work. And at the end, they blamed it on the biomarker. But that can happen with any biomarker. So be sure if you use your test for changing therapy, that the other therapy also has a chance to work that you switch to. That's a very important message that we learned. So at the end, a big thank you to all the people here in the Institute. You see that the sun is shining in Hamburg, actually now for 6 weeks in a row. Michael, it's incredible. We cannot believe that we are still in the north of Germany. So we are happy for the climate change there, you know. And of course, a big thank you for the funding organizations. And thank you very much for your attention.